Thank you, David.
Yeah, thank you for coming. I'm only going to use a script for the first few minutes, and then I'm going to completely ad-lib. I think it's more natural that way. So first, let me thank King's Church for inviting me to share with you this evening. My particular thanks go to Richard, who made a fantastic job of sorting out the slides for me. Last year, you had a lecture on the really big objects in the universe. And this year, I shall be looking at the other end of things, the tiny end of creation, as we take a look at bacteria and even smaller objects like chemical molecules. And if we ask the question, and everybody does at some point in their life, how did life come into existence? There are basically two possibilities. Either creation has a creator, which to me sounds logical, or alternatively, amazing things can happen by accident. And if I tell you how I became a scientist, you'll understand why I want to mention chemistry and biochemistry as well as bacteriology. Even though I was primarily a bacteriologist, I actually published more papers in the chemical journals than some lecturers in the chemistry department. This book, for example, which was published 41 years ago, is actually on chemical analysis technique. It's a nice book, except for one thing which is still shocking. How much do you think a book like this would have cost 41 years ago? And the answer is, 41 years ago, it cost £42. And I could be overseas at a conference and I'd have people coming up to me saying, I'd love to buy your book, but I can't afford that price. So it was bought by university libraries. I want to start with when I was a primary school child. At that point, my only career aspiration was to become a locomotive driver. And after decades, I discovered recently how much they earn, and I realized I may have made a mistake after all. And as I stood next to this shed, I had no idea how important it would become in my life. At age 11, I progressed to a grammar school, and for the first time I encountered chemistry, which absolutely fascinated me. Right, that's me. Now, that boy called John was the most violent boy in the school, and you know him because his name was John Lennon. And someone once said to me, he must be a lot wealthier than I was. Well, he was, but at the time he'd been shot. And I said, yes, but I'm more alive than he is. Well, aged 11, I think that Christmas, I had a chemistry set from Father Christmas. That was nice. And years ago, it was normal for youngsters to do chemical experiments as a hobby and inevitably also experiment making explosives. I don't think you could do it nowadays. My bedroom also served as my laboratory, and aged about 13, I was allowed to expand activities into the kitchen, where I decided to heat together some finely powdered zinc metal and sulfur. This was to make a chemical called zinc sulfide, and the test tube, as I heated it, and as the contents melted, it took on a life of its own. It emitted a bluish-green light, and there was a whoosh, and the molten contents passed my face and landed on the ceiling. And our ceiling remained the only ceiling in the UK to have a scab of zinc sulfide on it. And I learned some years later on that NASA had been testing the same chemicals as a potential solid fuel propellant for rockets. And this was the point when I was banished into the garden shed, and that was turned into my laboratory. And the shed later witnessed another milestone in my life when, for the first time, I kissed the schoolgirl who eventually became my first wife for 45 years. And nowadays, I do not kiss schoolgirls in sheds. In my teens, I also became a Christian. And it wasn't in the shed, and it wasn't actually in a church building. It was in the house of a wonderful Christian couple who amazingly kept open house. Any night of the week, teenagers could go along and they would give them cups of tea and biscuits and be very friendly and kind towards them. On a Sunday evening, first time I went along was a Sunday evening, they were having a Bible study and they were looking at the Sermon on the Mount. The man in the family was a man called John Rogers. He and his wife had Gladys Aylward stay with them, and she was, the older ones will know, as a missionary in China. She had a Hollywood film made about her life. And John Rogers, as a young graduate, had also assisted a very maverick Church of Scotland minister called George MacLeod, who later became Lord MacLeod, as he restored the Iona community. So on my first evening, I met this very charismatic man, and I remember being very surprised by having the Sermon on the Mount explained to me. And I asked to see him a couple of days later, and that was when he explained to me, no, you don't get to heaven by being good, which is what my mother had always told me, but by making Jesus Christ the center of your life. And that altered my whole outlook on life. And a few months later, this couple had a baby. When the baby arrived, the experts said in the hospital, it's only got 2 hours to live. And everyone connected with that fellowship and that family was asked to pray. And at the time, I remember thinking, well, if the experts have said the baby's only got 2 hours, why are we bothering to pray? But a miracle happened, and the miracle, it's not me saying it's a miracle, it's the people in the hospital. The child lived. And that was when I realized that the Christian faith is far more than just a logical argument or even an ethos. And some years later, well, about the next year it was, I entered the sixth form and I studied physics, chemistry, maths, and zoology. They didn't actually all fit in the timetable, so the first year I taught myself half the A-level chemistry. And I really annoyed people in the upper sixth form because I won the school prize in chemistry. And the next year, I had to teach myself half the physics course, but I actually did pass physics. And I went to university to study biochemistry, went to Liverpool University. At school, I'd been told in my first week in zoology, I'd heard about the theory of evolution, and when you're 16, you tend to think schoolmasters know best. And it actually took me a long time to realize that the fundamental problem is not evolution, because until you've got life, nothing can evolve anyway. The big problem is creation itself. How did life start? How can simple chemicals rearrange themselves into unbelievably complicated living cells? And 60 years ago, I believed that everything would become clear as more and more scientific discoveries were made. And although the complexity of the universe has become clearer and clearer, what's become more and more puzzling is how on earth it could have happened in the first place. When I was studying biochemistry, which also included chemistry up to degree level, we had a fascinating guest lecture on bacteria, and the lecturer explained they can do things we can't do, including some bacteria can actually consume Dettol. And after I completed my biochemistry course, I then did a bacteriology course. The courses overlapped, which is why it didn't take too many years. And when I started, I thought there would be just a handful of different bacteria, shaped like sausages, filled with jelly, very simple. Well, how wrong I was. And I remember going into the university library one day, and I thought, wonder if I can find one of the oldest journals in the library, and it was a copy of Nature. And I read an article by a very famous biologist in Victorian times, Huxley, and he wrote, "I confidently predict that in the next 10 years, life will be created in the test tube." And over 140 years later, I'm still waiting. Now, last year's lecturer had slides of himself with Stephen Hawking and with the Pope. Well, I've met neither, and I haven't gone around having photographs made of myself, so you'll just have to take my word for this. But I have done research with the leading dental scientist in Australia, Greg Seymour. I've done research with a chap called Bob Fitzgerald in Miami, who was the man who discovered the main bacteria that cause tooth decay. And I've also done research in Boston, Massachusetts, with a lady called Ann Tanner, who discovered which species of bacteria are principally responsible for gum disease. And I also collaborated with other world-class researchers. And one highlight was being the guest of Professor Bengt Gustafsson at the Karolinska Institute in Stockholm. He was the chairman of the Swedish MRC, the Medical Research Council, and he was also secretary of the committee that chooses the winner each autumn of the Nobel Prize in Medicine. And I have to add, although he came back from his yachting holiday early, especially to see me, I was never under consideration for a Nobel Prize. Let's get that clear. Well, I want to show you some slides, and they'll illustrate the wide-ranging abilities of bacteria. And I'll even give you some information on which ones to use as weapons of mass destruction. Not that I expect you'll want to. And I'll also try and mention, it'll just be very briefly, some of my own work. And I also want to look from a chemical point of view at some of the problems of life just creating itself. And finally, if time permits, I just want to add a bit more from a personal point of view. So let's move on. This slide is an interesting one. It's the oldest drawing we have of bacteria from hundreds of years ago, and it was made by Anthony van Leeuwenhoek, a Dutchman who was looking at water in Dutch canals. So the canals weren't clean even then. Now, this man is a real hero of mine, Louis Pasteur. He had arguments that he won, and strangely, some people are still wanting to have them now. Some people believe life could just happen. They believed in spontaneous generation of life. Louis Pasteur did a lot of experiments to prove those people were wrong. But he was an amazing man. He worked out how to immunize people against rabies, and it's only fairly recently that better vaccines have been available. He invented a whole field of chemistry, stereochemistry, which I'll be actually mentioning tonight. What else did he do? He invented something you all have used or benefited from, pasteurization. And he invented it. This may surprise you. It wasn't to make milk safe. It was to stop wine souring when it was sent around the world to the French colonies. And of course, he's known as the father of bacteriology. So he was very interested in which bacteria were causing which diseases. This slide shows what happens under absolutely ideal conditions if bacteria are growing absolutely flat out. The fastest ones can multiply to such an extent. To say they increase tenfold in an hour is probably a very slight exaggeration, but it keeps the arithmetic simpler for me. But it's not far out. You know, after 6 hours, one bacterium can be 1,000,000. So this is why my wife gets told off if she leaves food outside the fridge, because I'm thinking even in an hour, we could have a lot more bacteria. Something else that's probably relevant to our daily lives is the temperature which bacteria can grow at. There are bacteria that actually are quite happy to grow in fridge temperatures. But the bacteria that I worked on that cause tooth decay, when they cool down to about 20 degrees, just stop growing. They switch off. So if they were in the fridge, they couldn't grow. The organisms that make us ill and parasitize us are mesophiles. Thermophiles are interesting. You've met them, but you haven't ever realized. In the days when people used the washing machine at a high temperature, remember when bio-detergents came out? Well, normal enzymes would have been inactivated by the high temperature, and they had to get the enzymes from thermophilic bacteria. So bacteria that could grow at a high temperature had enzymes that would be happy at high temperature. The extreme thermophiles will grow even in boiling water. And this picture just shows a typical example. Apparently, a human being disappeared in this pool. They never found his body. But it would almost certainly have had bacteria growing in it. Now, there are bacteria that won't stand high temperatures, and the cells will die. If you heat them up to boiling water temperature, you'd kill the cells. But what you won't kill are some very heat-resistant spores that the cells make. And these spores are one reason why in the canning industry, well, and also things like fruit bottling, you have to be very careful to kill the spores. And this can be done by using higher temperatures than 100 degrees. If you were using a pressure cooker and you'd be up to about 120 degrees, that will even kill the spores. How tough are bacteria? Well, I've already mentioned that some can resist Dettol. In my first lecturing job, after I left Liverpool, I went down to Cardiff, and when we wanted students to have one particular organism to use in a laboratory class, the technicians used to go into one of the clinics, go to the wash basin, put a swab down it, and they would end up with a pure culture of one particular organism, even though things like Dettol were being poured down the sink every day. Yeah, some bacteria resist drying. There are even some that resist radioactivity, and you find these in where there's a higher background level of radioactivity near a power station, for example. Some not only resist acid, there's one species that actually produces sulfuric acid, and it can actually rot concrete pipes. So this is why I say they are just amazingly different from one another and from us. Some we know are very resistant to particular antibiotics. And back in the 1960s, it was found that bacteria were resistant to antibiotics they'd never even met. And the reason is bacteria that had met the antibiotics outside their chromosome had other DNA, and that could be passed from one cell to another, and it passed the information over. And at the time, I remember thinking, well, at this rate, no antibiotic will be any use at all in a handful of years. Fortunately, a lot of antibiotics are still useful. Do bacteria need oxygen? Well, there are some bacteria like us. They can't grow without oxygen. At the other end of the scale, there are bacteria that I worked on. If they're exposed to air, just with 20% oxygen, for 30 seconds, even if you've got millions of the cells, you'll kill them all. So we had to grow them in special apparatus. And this apparatus would be, in old measurements, about 5 feet wide and about 3 feet tall. It would sit on top of a bench in the lab. And to put things inside the apparatus or take it out, there was a double door system, a bit like a double door system on a submarine. And you wouldn't open both the doors on the submarine at the same time, and you wouldn't open both of these at the same time. And you had to suck out all the air and replace it with gas without air a couple of times before you could even think about opening the inner door. This slide is actually a really important one because it shows the complexity of bacteria. I've seen estimates of somewhere between 20 and 50 different genes are needed to code for components of this structure. A bacterial flagellum is used to push the cell through the water. And when I was an undergraduate, I was told flagellum, which means a whip in Latin, I was told it worked a bit like cracking a whip, and you had a ripple effect going down, pushing against the water. And about maybe 30 years ago now, someone said, no, it works a bit like a motor with a propeller. The flagellum goes round. And the motor, the flagellum, I need to explain, if you think a bacterial cell is about 1,000th of a millimeter in diameter, this flagellum is just a few millionths of a millimeter in thickness. And the motor that turns it round is similarly, there are components there that will only be a few millionths of a millimeter, and they will contain lots of different proteins, and proteins are not simple molecules. They can be quite big molecules. They each have to be coded for by DNA. And it's just mind-boggling to me, boggles my mind, that you can have this structure so small spinning round in a bacterial cell, and it's spinning round just in the outer layer of the bacterial cell. Imagine an orange and think of the peel. Well, bacteria don't have a peel, but this structure in this slide has got 3 layers. But it's much thinner than the width of a bacterial cell, just so small. And there are people who don't want to admit maybe creation does have a creator, and they say, well, some of these proteins actually already exist in bacteria. Well, my answer to that would be, you still haven't explained how they got there in the first place, but what about all the proteins which don't have any other known function? Because the idea of evolution, as I understand it, is you keep selecting the fittest. And if you have two bacteria, one of them is making lots of proteins that can't do anything because it still lacks certain proteins to make this thing work. If you have the two bacteria, one hasn't got any of these, it doesn't have a flagellum, doesn't have any of these proteins, so all its manufacturing capability is spent doing things that are useful for the cell. The second example, a lot of energy is being wasted all the time, and it would have to be for thousands of years making something that's no use. Natural selection, to me, makes me think it's the cell that isn't wasting time making things it doesn't need that will get selected. So that would work against this ever developing to the stage where you've got a working motor. This slide is interesting. The red line, if you just look at the red line on the graph, it shows the population of this country. I've seen similar data for the whole of Europe, and it shows what happens with the Black Death. It took 250 years for the population to get back to where it had been, and the decrease in the population was the spread of Black Death. And that's caused by an organism called Yersinia pestis. And the Germ Warfare Defense Establishment, when I was identifying bacteria chemically, thought this would be a very quick way. If someone attacked this country, my techniques could pick up the organism quickly. And I was nearly tempted to let them give me lots of research money. And I suddenly thought, if this organism escapes and I wipe out half the medical school, how am I going to go along to the Vice-Chancellor's office and say, there's been a slight mistake, I'm sorry. So I didn't work with them in the end. Tetanus is caused by one of the spore-forming organisms I mentioned before, and it has a toxin that has this horrible effect of contracting muscles. Muscles go into spasm. Now, I've actually been protected against this, and probably a lot of you will have been, by being immunized with tetanus toxoid. The toxoid is just the toxin made harmless, but the body can make antibodies to it and protect us. Botulinum toxin, again, it's made from another of these spore-forming bacteria. And at a conference in America once, a friend of mine was giving a talk on his work on this toxin. And he had a lovely slide. I wish I could have had it to show you. It was a photograph of a newspaper. And it was when weapons of mass destruction were in the news. So the whole front page of the paper was about weapons of mass destruction and telling people how terrible this toxin is. And it is 1,000,000 times more poisonous than cyanide. But in the corner of the paper, there was an advert, and it was for Botox for ladies to be more beautiful. And that was the same thing, but more dilute. This picture shows somebody who's suffering from a skin lesion caused by anthrax, and you can see under the microscope you have this sort of chain, like a chain of sausages, it looks like. So they're the bacteria that are causing anthrax. Anthrax, again, has these very resistant spores. And experiments were carried out a long time ago. It may have been at the time of the First World War. They actually deliberately infected an island off the coast of Scotland. And the same person who wanted me to risk wiping out half the medical school, Richard Manchie, had the job, and he was on the national news, he had the job of making the island safe. So he had to go up to Scotland and treat the whole island with disinfectant to try and get rid of any trace of anthrax. This shows the island. And an older picture shows people in protective suits and warnings telling them people not to go there. Now this, again, worries people, weapon of mass destruction. This smallpox isn't caused by a bacterium, it's caused by a virus. And people of my generation who'd been vaccinated, this is where vaccination comes from. Vaccinia was a virus used to immunize people against smallpox. Yes, people of my generation were very well protected by being vaccinated. But I was thinking the other day, years ago, health and safety really wasn't very good. I can remember working with smallpox virus in a lab, no specs, no gloves, no mask, the door open, people who may not have been immunized walking past the lab. And there were two of the lecturers in that department went to other universities. One went to London, and the virus escaped from his lab and put someone in hospital, but he survived. And sadly, the other one, who became a professor at Birmingham University, had the virus escape, and it killed someone. And the local newspaper launched a kind of hate program against him and made his life a misery. And one Saturday, his wife had gone out to the shops. She came home. He wasn't in the house, and she went outside. And when she went into the garden shed, he was just lying in a pool of blood. He slashed his throat. And nowadays, the smallpox is, I think, kept in a lab in Russia, in the United States. I'm not sure if there isn't a lab still in Europe somewhere, but it's been carefully tucked away so it can't be used to wipe out populations. Now, this is an interesting picture. I was actually the first person in history to look at bacteria with a scanning electron microscope. So many years ago, it must have been, I think, 1969, I and a colleague in Cardiff were able to publish pictures of bacteria growing on a solid surface, magnified 20,000 times. And these pictures got into most of the microbiology student textbooks of the time. This organism is one that is important in things like cheese making, but it has at least one species that's important in producing tooth decay. Now, the next one, this actually isn't a bacterium, it's a yeast, and it's bigger than a bacterial cell, and the cells are not all the same size. It can cause problems for human beings. It can cause something called denture sore mouth. But if you had this in your bloodstream, it really wouldn't be very good for you. Now, Streptococcus mutans is an organism that was first classified properly because of the PhD study I did. And other people, as I say, had discovered this organism. One person in Sweden had suggested you could call it this name, Streptococcus mutans. But we tested over 250 organisms, and we used about 80 different tests. And we had the problem, we couldn't find a computer big enough to crunch up the data. So we had to go to ICI, and the management services section did have one computer big enough, and they crunched up the data for me. And it's interesting that I recalculated the data about 30 years later in a laptop. The original calculation had taken this huge computer something like half an hour. and the laptop did it in about 30 seconds. Now it would probably do it in a fraction of a second. And I was looking recently to see what the internet says about tooth decay. And it's interesting, it does say that the main organism is Streptococcus mutans, but that people without that organism will still have tooth decay. And I spent about 20 years discovering which other organisms could. And the Medical Research Council funded that research. And although we found more organisms than anybody else, scientists, as I discovered, are not very open-minded and fair-minded. They can be very bigoted. And even the person who discovered this organism, Streptococcus mutans, Bob Fitzgerald, told me once he was really frustrated. He'd used the same techniques to find a species of lactobacillus that also caused caries. And although everybody accepted the research he'd done for the Streptococcus, they didn't want to believe what he's found with lactobacillus, which is ridiculous. But people are not very open-minded, and they should be. Well, now I'm going to pretend to be a chemist again. Miller was somebody who got the Nobel Prize for chemistry. He wanted to find out, how do you get some of the basic building blocks you need for proteins? They're called amino acids, and there are about 20 of them of relevance. And he just took some gases, the ones he'd been told were in the atmosphere long, long ago. So things like water vapor, hydrogen, methane, ammonia, and he actually did produce some amino acids. Now, the only problem is producing amino acids chemically actually doesn't work if you want to create life, because amino acids, with a single exception, come in two versions. It's like a left-hand drive car and a right-hand drive car. They're similar, but they're not the same. And what you want for proteins are, if you like, the left-hand drive version. But if you've got a mixture, you can't make proteins, because the very things that would sort it out in a laboratory now are enzymes, and enzymes are proteins, but proteins don't exist, remember. This is going back in time. So we're still left with a huge problem. Another problem, and this was found in Miami by somebody called Dr. Fox. He did similar experiments, and he published a paper. He'd made the simplest possible amino acid. A year later, he published another paper. He'd now made the second most simple amino acid. And a year later, he published another paper. And he had to admit that what happened was, instead of getting all the other amino acids, the ones he'd already made started reacting with one another. And he ended up with just a mess. So even though it's fascinating to think this experiment, putting in some energy with electrical spark, a bit like a bolt of lightning, it sounds good, but there are huge problems when you actually look at it. I've mentioned about the amino acids being, if you like, left hand or right hand, and this actually gives a model. Imagine this is a little model. So you've got this orange ball, the blue one, purple one, the black one, the white one. If I had those models here tonight and said, I want you to turn one round to make it the same as the other, you couldn't do it. It's physically impossible, or it's intellectually impossible, because one of them is like a mirror image of the other one. And this is coming right back to the discovery made by Louis Pasteur. So we have this problem, and I've not come across anyone yet who can explain to me how just by chemical means you can just make just one version of each of the amino acids. Now this slide, this is actually a tiny bit of this chart. The chart I've got here, which you're welcome to have a look at, it's covered with chemical equations. And those equations give you some idea of some, but not all, the chemical reactions that go on inside bacteria. And they're very similar to the ones that go on inside us. Bacteria can do other things which are a bit cleverer. They can make acetone, which you use for taking nail varnish off. Don't use it myself. Have used it myself, though, doing fiberglassing. They can make other chemicals which can be turned into solvents in paint. There's another chemical some of them make, which is, can be used in the screen wash as antifreeze in your screen wash in your car. So if they didn't make that from the petrochemical industry, bacteria could make it anyway. So they're very clever. And this slide shows how to make just one amino acid, glutamate. What it doesn't show you, there are some numbers in black, very small. Those are enzymes you need, which are catalysts that make the reactions take place fast enough to be any use. But those enzymes are proteins. And if we're trying to work out how life started, how you go from very simple chemicals when there aren't any proteins, how do you make any of these basic building blocks when you haven't got the proteins you need to make them? It's a real chicken and egg situation. Oh, this slide, this is also on this chart. And I hadn't really stopped to think about this before. There are two chemical structures, very, very similar. One of them seems to have a hole in the middle, but actually, instead of having an atom of iron in the middle, like the hemoglobin, this bacterial chlorophyll could have an atom of magnesium. And the two structures then are just amazingly similar. And I also discovered, although this is in the 22nd or 23rd edition, this map of metabolic pathways, there's a chemical mistake in the structure. So maybe someone thinks they're a chemist. Have a look at it afterwards and tell me if I'm right or not. Now this is the Eland Valley Reservoir, very impressive. And this is so we can start thinking about a basic problem you've got from chemistry if you want life just to make itself. If you've got a reservoir, water will go from a high level to the bottom, low down. But unless you pump it up, it's not going to go uphill. We never see water go uphill. And a fundamental rule seems to be things go from a high energy level to a low energy level. Something else we know, and I also know from chemistry, is this dog is actually, this isn't actually a dog, it's made from sand. Just amazing. Yeah, just made from sand. Even the little puppies feeding, they're just made from sand. But if we had a disaster, if I tripped over and fell, apart from the fact that the chap who made it might kill me, what would happen to the dog? Would it be turned into something that was more highly organized, or would the accident result in it being less well organized? And the answer is very obvious. It would be less well organized. Accidents do not tend to organize things. This is a basic problem. If you think life can create itself, life is very, very complicated. How can something become more complicated? Now, these slides, I haven't got time to go into them, but I said I became a Christian when I was 16. One day in my job, I had a phone call, and it was from somebody who was an expert on cot deaths. And he said I had techniques he needed. Nobody else had them in the country. He wanted me to help him. And I nearly said, sorry, I'm just too busy. And then I thought, if parents lose a baby, that's absolutely terrible. I've got to help him. And fortunately, I did. When he came to see me, within probably 30 seconds, I was thinking, this guy's a genius. And I worked with him for many years. And we did a lot of work discovering which bacteria are important in cot death, which toxins are important, and then I went on to look at which genes are important. And this resulted in my ending up being an expert called by the courts in murder trials. I got involved in about 24 cases where women were either actually sent to prison or in danger of being sent to prison for murdering babies when they'd simply suffered a cot death. But there was one case that was the other way around. It was because I and another expert asked to see the coroner in a coroner's court, and we said, we think these papers should be referred back to the Director of Public Prosecution. And the woman we suspected had killed the baby was found guilty, and when she was in prison, she then admitted to another prisoner that she'd actually also killed the first child who died, who had been regarded as a cot death. So then she got a little longer in prison. And we started a little bit late, so go on just a couple of minutes. Although what I've told you tonight convinces me you can't explain life and just ignore God. Let me tell you two experiences in my life that are very difficult to explain simply in scientific terms. The first one about 20 years ago is when I had a problem and I just had a feeling, and it was just a feeling, I should go along to Cheadle Baptist Church. I'd never been there before, didn't know where it was, and my wife pooh-poohed the idea. She said, well, no, we should be going to our own church next Sunday. A few months later, the same thing happened, and she said the same thing again. A few months later, the same thing happened. Interesting, it was the third time. And that time, my wife said, well, actually, I can't go this Sunday, so yeah, you go. I didn't know where the place was exactly, so I left the house early, and I got there when there was only one other car in the car park. And when I went into the church, there was a woman who was fussing around who then disappeared. There was one man at the far side of the church, and he came over and spoke to me. And he explained to me, he didn't go to that church either. He was a member of a Pentecostal church miles away in the Peak District. And he said, God told me to come here today to meet a man. And when I explained a problem I was having and what I thought I should do, he affirmed me that my position was the Christian position. I cannot remotely explain to you scientifically how that happened. I just can't. And then another one, this was 13 and a half years ago, just before Christmas. My wife, a few days earlier, had actually swum a whole kilometre at the health club. She seemed to be in good health. And she was going out to a choir practice. And she called out, and I'm going now. And I called back, I'll see you later. And before I could finish saying it, the door shut. And I remember thinking, she won't have heard what I said, but it won't matter because I'll see her later. Well, later, I had an absolutely, when I say a frightening experience, I couldn't explain it scientifically, I think that's what terrified me. If someone came into a room and switched the light on, you would instantly have light. But supposing nobody came into the room but switched on violent wind inside a house. And what frightened me was, I looked at the door, it was shut. I looked at the windows, they were shut. There was no soot coming down the chimney. There wasn't any wind coming from there. And after about 30 seconds, instantly it stopped. And it was just very, very calm and peaceful. And I remember thinking, what on earth was all that about? And the phone rang, and I looked at the clock and I thought, oh, I bet this will be Tricia. She's probably got back to the car and it's got a puncture. She wants me to go and sort it out. And when I picked up the phone, it was somebody else who'd been at the choir practice, a friend called Gerald. And he said, Tricia's fallen over and she hasn't got up. And I said, what do you mean? And he said, she's fallen over and she hasn't got up. Her head's resting on my shoe. And I could hear tremendous racket in the background. The same evening as the choir practice, there'd been an event at church, and two of the people who were leaving happened to be medics, and I could hear them trying to resuscitate my wife. And Gerald, bless him, he realized I couldn't get there, so he kept his phone going so I could hear. And I remember saying to him, can you ask them, is she breathing? Does she have any heartbeat? And the answer was no. And I said, I think she's gone. Normally when people die like that, their attitude is, no, it can't have happened. I just knew she died. And Gerald said to me, I think you may be right. And people have said to me, and I know this is true, you can resuscitate people, but I just knew she wasn't going to be. And in fact, when she had a post-mortem, sometime later, it was found the thoracic aorta had severed, so they were never going to save her. And my feeling at the time was, well, coming back from the hospital later on, and I was thinking, my whole future's just smashed to pieces now. That is when God spoke to me, not like someone speaking to me, like you might speak to me, but just like almost like my conscience speaking to me. God said, you don't have to worry about Tricia. She won't be in your loving arms, she'll be in mine. And he said, you don't have to worry about yourself. I'll continue to walk with you, and I have work for you to do. And I had no idea what that was all about. 3 days later, I was setting up a charity that has raised over £40,000 to help people in poverty. And God has used me in all sorts of other ways. There are people here from the Knutsford and Alderley Edge circuit, and they'll know the way I pestered all the churches, not for money, but for things like clothing I could give street homeless people, things to give a children's charity. There are just so many things have happened. Currently, I'm taking food from the Methodist Church and the Roman Catholic Church and other individuals from Alderley Edge to a food bank in Wythenshawe, which is a huge council estate area about 7 miles away. And I'm not telling you that to prove how clever I am or how wonderful I am, just to prove how wonderful God is. He can use any of us. If he can use me, he can use you. Well, thank you for listening to me this evening, and I hope something I've said will be helpful to you. Thank you.
You guys do know there's a test. I've got to say that as I listen to that, you frightened me, because weapons of mass destruction are supposed to be things you can see. But what also came to mind was, with all these things around and how impossible it would be to defend yourself, we have a God who keeps things in balance. And that's all the proof I need that there is a God, because he takes the bacteria and uses it for good and protects us. And so, yeah, absolutely mind-boggling to recognize all these different things and to share part of your journey. But we wanted to give an opportunity for you guys to ask. I did say there were questions, and so yes, for questions. But if we can get you to speak into the mic, because we are recording. I'll come to you.
Thank you very much for that very stimulating talk, Dr. Drucker, or Professor Drucker. I have two questions. One, I suppose, is a little bit technical, and forgive me for that. And the other one is a much more general one, might be more interesting to others. So I sort of have a science engineering background, but I have never understood why proteins, etc., have to be levorotatory left-handed as opposed to dextrorotatory right-handed. I've never really understood that. Is there any way you can explain that for someone who sort of dabbles in this but doesn't really understand it? And if not, it doesn't matter. The next question is more important, I think.
Do you want me to answer? Shall I answer that?
Yeah, sure.
Yeah. Many years ago, people actually were clever enough to do things I can't do. They actually worked out the actual shape of proteins, because the shape, the overall shape, is what's crucial. There'll be like a little, for want of a better word, a sort of little docking station in a protein that's an enzyme, and the chemical the enzyme is supposed to be working on, that will fit it exactly, be like a lock and key. Now, if you have the wrong amino acid, the actual shape of the protein is corrupted. So it's almost like having a lock and key, but suddenly the lock is the wrong shape for the key to go in.
Right. Okay. Yeah, I guess, yeah, I have heard that before, but I've never understood why there couldn't be molecular structures that are sort of all working the other way and still working. So if the whole field, if you like, were
that's a very good point. If I can give you an example, not with an amino acid, with an acid that is made by bacterium, a dairy field, lactic acid. Lactic acid can come in two forms, L-lactic acid and D-lactic acid. And if I wanted to measure them separately, I actually use different enzymes. You can get an L version of the enzyme, you can get a D version of the enzyme, and they each recognize a specific mirror image version of lactic acid. So it's the same principle.
I see. The other question I think is probably more generally of interest. You said that quite early on, after talking, in fact, about lactobacillus, you said that you found by your own experience that science or scientific people or scientists are not very open-minded, which surprised you. Why do you think that is the reality?
I think partly because the human beings aren't. But when I worked in the dental field, there was a problem that I remember someone from London University commenting to me that people were absolutely vicious with other people in the field, and they wanted to do them down because there wasn't enough research money. It was just like the law of the jungle. Yeah, yeah. Things could get a bit vicious. And also, of course, people aren't very good at admitting if they got it wrong. With one organism I found very early on that could cause tooth decay, nobody was very interested for about 20 years, and I was at a conference I think it was in Vancouver, and there was someone from Japan who'd been doing research in Japan, and he was telling the world this organism caused tooth decay. And it was strange when I was trying to tell him, I know it does, I showed that 20 years ago. It was almost as though he'd put his fingers in his ears, he just had to blurt out his little speech.
It's interesting that when I first came into the ministry, it was explained to me that do not look at your fellow ministers as brothers and sisters. They are fighting for the same post as you've got. And it's true. I've seen that so many times. People are people. Other questions? Just raise a hand. Yeah, Jeff. Had to have a question from Jeff.
Yeah, a long time ago when I was at medical school, we taught about cannibals eating the brains of other people.
Yes.
And they developed a similar thing to like mad cow disease.
Yes, in Papua New Guinea.
Papua New Guinea.
Yeah.
We used to call it then slow virus. We now call them prions.
Yes.
And prions are not even viruses, are they?
No, it's strange. Mind you, when I was a student, people even argued, are viruses really living organisms? Because even viruses, which are more sophisticated than prions, they can't live on their own. They can only live as parasites of some other living cells. One interesting fact I didn't mention is there are viruses like smallpox that infect us. There are viruses like tobacco mosaic virus, which could attack your plants. There are viruses which actually attack bacteria. And people have even suggested if antibiotics didn't work at all, we could use some of those to kill bacteria instead.
Can I ask another question? We know about the microbiome. Everyone knows about that if they read the Daily Mail. But there's some research now saying that bacteria can produce like dopamine, and dopamine can affect your mood. So certain bacteria in the gut can affect your mood level. And they're saying that it's possible, we use it for other reasons, that we can do a fecal transplantation. You can take the feces that we pass, put them in a liquidizer, if you're a healthy person, and then give them by enema to someone who is severely depressed.
Or someone you don't like.
Yes. well, the Bible says better to give than to receive, isn't it? But they're using it for Crohn's disease, colitis, and they say that resistant depression may eventually be treated by giving a fecal transplant.
Yes. No, I think we recognise bacteria have a bigger effect on us than we really imagine. I mean, even when I was still working in the dental school in Manchester, people are starting to realise that the bacteria growing in your mouth could be affecting your general health, not just your oral health.
Have you found, since you accepted Christ as your Saviour, the stimulation and the acceptance of what He's actually created? I don't know how to actually verbalise it more closely.
I think I know what you mean. I can marvel at what God's done instead of thinking, gosh, this is a cool accident.
This isn't a deep and meaningful question, this one, okay.
This will be the one that floors me.
You were talking about the enzymes that cause cavities in teeth, etc., that you did your study on, or bacteria that cause
the bacteria, yeah.
As a consequence of that, has there been anything that has counteracted that can reduce cavities in teeth?
Well, years ago, the Medical Research Council funded 2 research groups in London who were trying to make vaccines. And I was at a conference in Copenhagen, and one of them was giving a lecture, and when they had the questions, I asked the question, if dental caries killed people and the vaccine killed just a few, but less, it would be justifiable. But I said, people aren't dying from tooth decay. Your vaccine could kill them. How do you justify that? And the professor's answer was, sorry, I couldn't hear your question. And I thought, I'm sure you couldn't. No, I mean, all sorts of things obviously have been experimented with. And it always amuses me the way there's always a fashion for putting something in toothpaste. When I was young, toothpaste was green because it contained chlorophyll. Well, what does chlorophyll do? But I can tell you a funny story. A friend of mine worked for Unilever, which obviously makes toothpaste. And years ago, it was fashionable for people to create an artificial mouth. So you try to imitate an actual human mouth in the laboratory. And Unilever had a complaint from Sweden that their toothpaste was actually attacking teeth. So my friend had to fly off to Sweden, and he found out what the problem was that the artificial mouth had plastic teeth, and the toothpaste contained traces of chloroform, which dissolved the plastic.
I'd like to know, how do you get candida? A friend of mine had candida, and she was told it was like a mushroom that was growing inside her. And then it was eating all the stuff that she was eating, like sweet stuff. And she was taken into hospital, and she had to have poison dripped into her body to try and kill the mushroom-type candida that was in her body. She's never really got rid of it, but she can't have anything sweet, or she's hardly anything that she can eat through having this candida.
Yeah, probably. Probably I would guess between 5 and 10 people in this room have it in their mouth now. So a lot of people will have it, but it won't be making them ill. The problem is if it gets into the bloodstream. In fact, in general, if microbes in the body get into part of the body that they are not normally present in, that's when you have a problem. There are lots of, organisms that cause gas gangrene live in our gut, but if they stay in the gut and the gut's healthy, they're not making us ill. But if you were fighting in a war and someone shot you and a bullet went through your abdomen, that's when you could end up with a serious infection.
Folks, we're going to have some coffee, because coffee's good. There's tea as well for those who don't drink coffee. Are there people like that? And an opportunity for you to come and chat to David in person or chat to somebody else. David attends our Knutsford church, so you can always grab him on a Sunday evening and say, I've got more questions. Right? But we really just appreciate, because you've opened us up to a totally different aspect of God's amazing creation. Right? And so let's give him a round of applause. We will be having another lecture in a couple of months' time. I'm not sure who. We're waiting on the Lord. But I just love this because there's just such a range. And maybe that person's here. And they've got something that they can share about faith and their own discoveries. So come and chat to me afterwards if you are that person. But we want to just find the most amazing and interesting people because they've got a story to tell. God bless. Thank you.